This is Professor Doherty, and today what I'm going to be going over is the second lecture on muscle physiology. Today's lecture should not be very long. There's not a lot of components to this part, but what we'll do is I'm going to show you real quickly a review of the sliding filament theory. So in our last lecture, we went over the sliding filament theory in a pictorial format. Today what I'm going to do is just show you the written version of it, so you can write that down if you like. Then we'll talk a little bit more about neuromuscular junctions, and then we'll take a look at twitches and how they lead to these graded muscle contractions eventually, and then a few muscle disorders, especially in regard to how different types of poisons can affect the functioning of the muscles. So first, sliding filament theory. Here, I'm just going to bring these up real quick, are the steps in the sliding filament theory. I'm not going to go through all of these again. This is basically just a repeat of, like I said, the drawn format that I gave you for the sliding filament theory. Here I'm just laying it out in words and steps. Now there is one additional step that I want to review because there are some special proteins that I did not talk about during the first sliding filament theory. And the reason I didn't is because they are different depending on what type of muscle we're looking at. So again, I'm continuing here with our theme of skeletal muscle. It's what I primarily focus on in this series of lectures. And so let me describe what we're looking at in this picture, and then you can write down some of the details. So you can see over here this small inset picture. It looks like the picture that we had from the other day. And we actually basically drew this picture with a little more detail. If you look here, you can see that this is the neuron, the motor neuron, coming down and making a neuromuscular junction onto the sarcolemma of the muscle cell. So this is the sarcolemma of the muscle cell. Here is the T-tubule, and then the green parts within this picture here are the terminal cisternae of the sarcoplasmic reticulum. Okay, so this is sarcoplasmic reticulum here. So if we move to this larger picture here, this is just a larger version of this region of this picture. So what we're looking at here is the sarcolemma of the muscle cell, and then here's the T-tubule. And now we can see in greater detail some of the proteins here that are actually involved with moving the action potential signal over to the sarcoplasmic reticulum in order to get the release of calcium from the sarcoplasmic reticulum. And again, this section here would be called the terminal cisternae. These are the parts that lay right next to the T-tubule. They form part of what's called the triad. So let me bring up a pen here, and I might change my pen color to something that might be seen a little better. Let me try a dark color. All right, so imagine here that there's an action potential moving along the cell surface here. And I'm actually going to, move my pen over here a little more so I can use a different pen. Alright, so imagine that here there's an action potential moving along the cell surface. This action potential was of course started at the neuromuscular junction, and so this is the request for a contraction of the muscle. This action potential moves along the cell surface and down into the T-tubule. It will also actually continue over here as well. It moves into the T-tubule until, well, and actually will continue down the T-tubule, but you can see in here a bunch of these purple, so I'll highlight one here, circle one here, these purple proteins. These purple proteins here that sit in the T-tubule are called dihydropyridine. We abbreviate that as DHP, so that's what you need to know. So dihydropyridine. These DHP receptors are voltage gated. All right, so you can probably write that down as well. They're voltage gated, meaning that the action potential, which is a voltage potential, will trigger them into action. All right, and so this action potential I'm going to show continues down this way. So when this action potential strikes these purple DHP proteins, they are voltage gated and they change conformationally. That's what happens with all of these proteins when they get hit with a voltage, as long as they're voltage gated. So in other words, they're going to twist a little bit. Imagine them as like twisting when they get hit with this voltage, okay? The DHP proteins in this case are not channels. Instead, they are directly linked to these yellow proteins behind them. I'm going to circle one here. You can see that there are two DHP proteins linked to this purple protein here. Or excuse me, linked to this yellow protein here. Notice that they are linked together, and this yellow protein is called a ryanodine receptor, also abbreviated as RYR. The DHP protein and ryanodine proteins are linked mechanically. So the ryanodine protein is a mechanically gated protein. And let me describe that again to make sure that people get this. When the DHP protein, which is voltage gated, gets hit with a voltage, it twists. This causes a mechanical deformation to be transferred to the ryanodine protein because they're linked together. The ryanodine protein, when it receives that conformational change, that twist from the DHP protein, then opens its channel. And you can see that the ryanodine protein is sitting directly on the sarcoplasmic reticulum, and you can see that's what's being shown here. Let me see if I can get a highlighter going. What's being shown here, oops, sorry, highlighter didn't work. Let me get that back. Okay, right here, there we go. All right, so you can see here the calcium is now able to leave. That's what this arrow here means, is able to leave the ryanodine receptor. You can also see the same thing here as well. So when a voltage hits the DHP receptor, it mechanically twists and twists open the ryanodine receptor, which is a mechanically gated receptor, allowing calcium to flow out into the rest of the cell over here. All right, so this is basically the step I kind of skipped, the details that I kind of skipped the other day because I wanted to go over them in a little bit more detail here. From the sliding filament model, that basically complete the sliding filament model. Now, why did I skip this part? Because this part will change when we take a look at smooth muscle and cardiac muscle. So here, again, what I'm talking about is skeletal muscle. So skeletal muscle, DHP and ryanodine proteins are physically linked together. All right, this is another look at the neuromuscular junction. So this is backing up to where the neuron meets the muscle. I just wanted to show this picture to show how invaginated the nerve is into the muscle. So if we take a look here, the top part of this picture, we can see an end plate that's been cut in half, so we can see the inside of it as well as the underlying muscle below it. Within the end plate, you can see that here we have the neuron that spreads out over the cell surface of the muscle a little bit. Basically, this is to give a wider area to be able to dump acetylcholine. Down here we can see the cell surface of the muscle here, so the sarcolemma. You can see how this turns and basically goes underneath these areas where the neurons are. So let's take a look at it at this inset picture here. This area here that I'm highlighting with my red pointer is the sarcolemma, and so you can see that the neuron is actually invaginating itself somewhat into the muscle cell, but it's not going into the muscle cell, it's just pushing down the surface of the muscle cell to make a wider area where it can dump acetylcholine. So remember, acetylcholine in skeletal muscle is going to be our neurotransmitter. You can see it here in these vesicles inside of the end of the neuron. They will be released into the neuromuscular or into the synaptic cleft here and then picked up by receptors on the other side over here. So I'm just showing you this to show you that these neuromuscular junctions are a little more complex than what we talked about before, that the broad end plate allows for more surface area to be touched by acetylcholine, and then also the neurons kind of push into the sarcolemma to make even more surface area contact. If we look at this in an actual micrograph, you can see here that here is a end plate So a neuromuscular junction is what that will form. Here is our muscle cell. Remember, our muscle cells look kind of like fibers, right? They're long. You can also see the extensions that go back on the neuron here for each of these. We can see another end plate and neuromuscular junction here. Remember that each one of these muscle cells, so here we can see maybe 1, 2, 3, maybe 4, 5, 6, maybe 7, a little bit of 7 here. These muscle cells will only have one neuron coming into them. So for instance, we will not see multiple end plates on this cell from multiple neurons. It's only going to be one. All right. So each muscle cell can only receive one neuromuscular junction or end plate. However, the end plates here may come from just one neuron. So in other words, a single neuron can go to multiple muscle cells, but a muscle cell can only have one neuron coming into it. All right, so now let's talk a little bit about twitches and graded muscle contractions. And so there'll be a few new terms here that I want you to know. Let's go ahead and go over to our drawing board here. And so again, this part is going to be describing certain types of muscle contractions, and we're going to start with twitches and then graded muscle contractions, because these are kind of related here. So let's talk first about what a muscle twitch is, because this is actually a technical term. A muscle twitch consists of a single end plate potential. If you remember, we described that as EPP. A single end plate potential, which what that means is that is a single neuron firing to the muscle, telling the muscle to contract. Okay, so essentially that's what EPP is describing. This is a single neuron basically dumping acetylcholine onto the muscle, which of course is going to tell it to contract. A single EPP happens in vivo, and what that means is in a live being. Okay, so this would be inside your body essentially. If you've ever heard of like vivisection, which is kind of a grotesque term in some ways, in that vivisection, which is using the term vivo here, means dissection of a living being. So for instance, like if you've ever dissected a frog, oftentimes what you'll do is you'll pith the frog, so essentially kill its brain by pithing it, and then do a live dissection. That is a vivisection. So that means in a living thing is what in vivo means. So a single EPP will happen in vivo, so inside a living body, or it will take place due to a single electrical shock. Now, a single electrical shock would be in vitro. So I'm sure that you probably heard of in vitro fertilization. So that refers to something that takes place outside of the body, is what in vitro means. So in vitro fertilization is where fertilization takes place usually in some form of a test tube, right? Referring to like these test tube babies, they call them, or they used to call them. Here, in vitro again means outside of the body. So you could put, for instance, if we use the same frog paradigm, if you've ever taken a frog leg and then hooked electrical stim modules up to it, you can make the frog leg contract. You can do the same thing on yourself. If you've ever seen or ever been in a physical therapy unit, they'll use what are called TENS units, at least that's what they used to call them. And you slap these basically electrodes on someone and turn on an electrical current, and you can actually see their muscles contracting. You can make their muscles contract because of that. All right, so there are a few things about this muscle twitch. So a muscle twitch, let me be very clear about this, is a single neuron telling a muscle to contract, or a muscle cell to contract, or a single electrical shock being given to cause that muscle to contract or muscle cells to contract. If we increase the stimulation, so in other words, we start having multiple muscle twitches, so increase the stimulus frequency. If we increase the stimulus frequency, what happens are several things. So let's talk about what those things are that happen. The first thing that happens is you get an increase in twitch strength. And there is a maximum here, and so I'm going to say up to max, which means that once you get to the maximum twitch strength, then you start moving into the next phase. The next phase, moving up from a twitch, is called summation. This is also sometimes called incomplete tetanus. And what this is referring to is when basically there's so many twitches that the muscle cell never fully relaxes. Okay, so this is when a muscle fiber is unable to relax between twitches. So in other words, now your stimulus is so frequent that your muscle cell is not going to back down into relaxation like it normally would after just a single twitch. Finally, in the next step, we get into, so summation leads to tetanus. And tetanus refers to a loss, and when I say loss, I mean complete loss of relaxation between stimuli. And again, here I'm still talking about the muscle, so basically what we're doing is we're moving up as far as contraction strength goes, okay? So in other words, a bunch of twitches lead to summation, and then an increase in summation up to the point of where you completely lose any relaxation leads into tetanus. All right. So basically what I'm showing you here is that there's a progression of increasing continuation of contraction. Now, if you hold tetanus for long enough, this leads to fatigue. And fatigue refers to a loss of force generation. So basically, eventually tetanus will decline due to fatigue if you hold that contraction long enough. And of course, we've all experienced this. If you ever try to, for instance, do a pull-up and just hold yourself up in that position, you'll notice that you'll only be able to do it for a little while before slowly you have to let yourself down, and that's due to muscle fatigue. So what I'm going to do here is draw a picture of what we just wrote out. So I'm going to make a little chart here. Down here we have time, and over here we have contraction force. So I'm just going to put contraction here. Imagine here that we have the baseline of a muscle. So this is the electrical activity of a muscle and its contraction, associated contraction at 0 essentially. So you can imagine that this is 0 contraction here. If we apply an electrical stimulus to it just very quickly, what we'll get is a contraction force going up and then dropping back down. All right, and we'll return back down to our baseline. This is called a twitch, and this is what it looks like if we look at it on a graph. Now this twitch, if we decide to send a bunch of electrical stimulation signals to the muscle, what you'll get is you'll get a twitch, and then you'll get a slight decline while the muscle tries to relax, and then we'll get another signal for another twitch, then another signal for another twitch. So it's trying to relax, but it's not able to fully relax. These, when you get something like this is called summation. So you can see that the difference here between a twitch and summation is that with summation we're never going back to our fully relaxed format. right? We're never going all the way back down to baseline. Now, if you get enough signals coming in, what will happen to the contraction of the muscle is it will become steady. And all of a sudden you'll lose any part of the waveform here that tries to go back down. And instead you get this summation essentially that goes to its peak. Okay, and you'll be able to hold that for a while. This is called tetanus. So the difference between tetanus and summation again is that notice that summation you have some drops between the twitches trying to go back down to resting. In tetanus you get a complete loss of that relaxation, so it's just a continuous contraction. Now if you hold tetanus for long enough, eventually the muscles and nerves will fatigue and you'll get this drop down, and so this sloping drop here is fatigue. There's a variety of reasons for fatigue. A lot of it has to do with the action potentials on the muscles and the imbalance of sodium and potassium that occurs from that. But there's a variety of other ways that you can get fatigue also. Okay, so with that, that basically is my first description describing tetanus, summation, twitches, summation, tetanus, and fatigue. So let's now move into the second part here. The next thing I want to talk about is sarcomere length. So what do our sarcomeres look like during a contraction, and where do we get the most force from? So remember, sarcomeres are our base units within the myofibrils, and remember that they run from one Z disc to another Z disc, and sarcomeres are the things that actually shorten during a contraction. Okay, so let's take a look at the length of a sarcomere. So sarcomere length is considered to be at 100% when it's at its kind of middle distance. So this is its ideal resting length. And at this length we also get maximum tension or maximum contraction, however you want to say that. So basically maximum force at this length. Let me draw Y here. Okay, so let's take a look. Here is a sarcomere. So these two lines here represent the Z discs, right? So that's one Z disc here, there's another one on the other side. Here is actin. And here is myosin. Okay, so just as a reminder here, green is actin and the purple is myosin. Now at this length, so this is the normal resting length, we actually have the maximum amount of force that we're going to be able to generate. And this has to do with the amount of area that is, and actually I am going to draw this out a little bit longer, the amount of area where myosin and actin overlap. So in other words, where you're going to get the maximum amount of tension is when we have the maximum amount of heads from the myosin able to grab onto, so I'm drawing some myosin heads here, able to grab onto the actin. Alright, so not going to do all of them here. So in this case we have an area, like this, where we have tension happening, or where we have actin and myosin able to grab onto each other. Now I may have drawn this a little too far out. There's probably more area than this that we would want to have touching, but hopefully you're getting the point. There's basically a point here where we have the maximum overlap, or the best overlap, between myosin and actin, and we call that length the 100% length, or ideal resting length. Typically, like I'm saying, this is at a kind of medium movement of the arm, or of any muscle, for instance. So for instance, As you're standing with your arm down to your side, let's use your bicep as an example. If you were to cock your bicep, maybe a little bit up from it where it's just sitting at your side, that's probably about where your maximum tension is at. You're getting the best overlap between actin and myosin. All right, the next type of length is the length of the sarcomere when it's between 0 to 80% of its normal length. Okay, so in other words, here this is a shortening type of phase, so it's been shortened here. Here there are some problems with this overlapping, so we're going to lose strength, basically, is what I'm saying. So this has less strength than ideal. And here are some of the reasons why. We get a massive overlap of actin with myosin. And because of this, we get fewer cross bridges. So let me draw this here. So imagine now our sarcomere is completely contracted like this. We have here our myosin, you can see, and what happens is the actin then overlaps on itself. And this actually decreases the force that's available for the actual sarcomere. So in this example here, our force generation has decreased. And what I want you to think of here, if we use the example of our bicep, if you were to take your bicep and fully contract it all the way up to the very top, what you'll find is that the force generated in that position is less than the force generated if you were to, for example, take your elbow down to a 90-degree angle. So at a 90-degree angle, you're actually getting better overlap of myosin and actin than at a full contraction. And of course, this leads us to our last part here. When the length of the sarcomere is greater than 120%. When the length of the sarcomere gets too long, we get almost no overlap of actin, and therefore almost no cross bridging. So if we look at that over here in a drawn format, what we would do is we would lengthen out our sarcomere to very, very long like this. So now it's being stretched. Our myosin still stays about the same length. And now our actin might barely be reaching our myosin. So you can see here now the overlap is maybe just this tiny region here, if anything at all. Now again, here we have much more loss of strength, so we again have less strength here than in our ideal position. Okay, and again, this leads to a decrease in force generation also for this scenario. And in fact, it's more of a loss than when we're fully contracted. So if we were to imagine this on our biceps again, if you took and you fully extended your elbow all the way out as far as you could, and then had someone push on your hand and try to pull upwards, back up to a 90-degree position, very, very difficult in that first part there, because there's almost no overlap of actin and myosin when you're initially making that contraction. Okay, so now let's take a look here back at our slides. And this picture here actually shows nicely the force generation that I was just talking about. So let's take a real quick look at this picture. I'll describe it for you here. This picture is showing basically power on this scale, so relative tension. So as we go higher here, our muscle is generating more power. And this is showing the percentage length of the sarcomeres in the muscle. Remember that somewhere around between 80 and 120% is where we get our maximum contraction. And so let's take a look at what happens when our muscle is fully contracted. So in this case, you can see here 1.65 micrometers is the length of the sarcomere when it's fully contracted. You can see the overlap of actin and how it's also still over myosin, but the myosin is now to the very ends of the Z discs here. They've been fully pulled in. And if we match that up to what's happening here, you can see that up here, our force generation has dropped off. And if we pull this in any farther, so this can contract a little farther because the myosin can actually squeeze in on itself, you can see that we quickly lose force. Okay, so as our percentage gets shorter, so as our muscle gets shorter, our power drops off precipitously when we get to the very top end of this range. And when I say top end, I mean the most contracted part of the muscle. If we look at the 2.25 micrometer here, which is about 120%, this is at the maximum part of the range as far as power goes because we have the best overlap of actin and myosin here. So the myosin heads can easily grab actin. There's no interference from overlap from the other actin. And so maximum power is generated, and this is in the middle range of that muscle like I was talking about. If this was the bicep, this would be somewhere around maybe the 90-degree point of the elbow. And then in the final stage here, and when we fully stretch out, let's say our bicep, and straighten our arm out all the way, if we try to contract that bicep muscle, you can see that we get very little power because we have little overlap of actin with myosin. And you can see that as we stretch this length up here, we get again a drop off in power until we go back down to zero at full stretch. Okay, so again, this is just showing how maximum power is really in a very short range in the middle of the muscle movement. Now you can actually see this at the gym sometime if you've ever gone to the gym and you ever see those big bodybuilder guys who are getting ready to bench press 350 or 400 pounds on the bench press, right? And you'll watch them and because they're usually making a lot of noise too, right? And what they do is they pick up the bar. This is what some of them do at least. And you expect them to do a full bench press, bringing it down to their chest. And instead what they do is they drop it like 2 inches and then they put it back, right? So they barely bring it down at all. And what they're doing there is they put on so much weight onto the bench press that only in their maximum overlap, so only when their sarcomeres are at their best position, can they actually handle that weight. If they were actually to bring that weight all the way into their chest, what would be happening is they would be stretching their chest muscles out like this. They would lose all this power and suddenly they wouldn't be able to lift that weight off their chest. So they only bring it down a short distance and then they push it back up and put it back. I don't think that that's a good way to weightlift, but that's the way that some people do it, at least when they're really going for power. All right, so besides power, or I should say another way to affect power is through the speed of the movement of the muscle. All right, and so that's what this picture is describing here. So basically here we have a force chart as opposed to the velocity or speed that a muscle is moving. Basically, here's the basic concept that you should walk away from this chart with, because it's a very simple chart. And that is that the faster that your muscle moves, the less force it generates. So you can see here that when you move your muscles or move your limbs very quickly, the force drops off very quickly as well. All right. You can generate maximum force by moving slower. And in fact, maximum force concentration is when you actually do an isometric contraction of your muscle. And of course, an isometric contraction is when you're not changing the muscle length at all. So if you change the muscle length very rapidly, it causes you to generate less force, which should kind of make sense because then the sarcomeres the myosin and actin are having difficulty grabbing each other quick enough to keep up with the shortening velocity. All right, next let's talk about some of the types of contractions. So you should go ahead and write this down, isotonic contraction types. We have two different types of what we call isotonic contractions. So the first thing here that you should note is that an isotonic contraction refers to a type of contraction where the muscle itself is moving. And what I mean by that is that the muscle is either contracting or it's lengthening. All right. So that is the overarching theme that we have for isotonic, is that the muscle is moving, either lengthening or shortening during the contraction. The first type of contraction here is concentric. And basically a concentric muscle contraction is when the muscle shortens during the contraction. So let me go back to the bicep example again. If you have some weights at the gym and you pull those weights up towards your head, contracting your biceps, so if you have the weights in your hand and you curl them, that would be a concentric contraction while you're lifting the weights up, because you're shortening the muscle length of the bicep. The other type of isotonic contraction is called eccentric, and this is when the muscles lengthen, and this is typically due to the resistance being greater than the force being generated by the muscle. So in our example, if you were weightlifting and you curled the weight up towards your head, that's concentric. When you lower the weight back down, that is eccentric, because there your muscle is allowing the weight to basically take the arm back down to its resting position. So you're relaxing the muscle enough to allow the weight to basically push it down. Now, just as a side note here, if you ever want to get really sore from a workout, then do an eccentric muscle workout, where basically your lifts concentrate on the downward movement of the muscle. For some reason, it makes your muscles really sore. I haven't looked into why. It's probably due to micro tearing, but all right, the other type of muscle contraction is isometric. So notice that this is separated out from the other ones, because an isometric contraction is one where the length of the muscle fiber does not change. Okay, so the fiber length is constant, and because of that, the load is not moved. Now, most isometric contractions that you do throughout the day are postural. So in other words, many of the muscles around your hips, in your lower back, and in your upper part of your legs are actually contracting as you're standing in place, just to hold you up and hold you in place. So an example of this, for instance, is that you have two muscles in your calf, right? Your calf muscle is actually two different muscles, the gastrocnemius and the soleus, for those of you who have taken anatomy. The gastrocnemius is more of a fast twitch muscle that's used for propelling you along. The soleus is more of a postural muscle. So in fact, when you're standing, the soleus muscle is actually slightly contracting to hold you up and to hold you in place. So these are small isometric contractions. Of course, you can also do this at the gym by simply taking a weight and holding it in one place for a period of time. All right, so lastly here, let's talk about muscle disorders, in particular how toxins affect muscles, and a couple different types of toxins here. So the first toxin I'm going to talk about is called botulinum. Botulinum is a poison that comes from a bacterium called Clostridium botulinum, and you can see a picture of it here. This poison botulinum is also known as Botox. And so I'm sure that you've heard of this because it's used in some cosmetic procedures, which I'll describe why they can use that in a moment. Now again, this is a toxin that comes from this bacterium. And what it does is it decreases the release of acetylcholine from motor neurons. So you can imagine if you get botulinum poisoning into your blood and it goes to your muscle cells, it will basically cause all your muscles to relax. It'll be difficult for you to move your muscles and move your skeletal muscles. Now, how is this used, and how is it used safely as a cosmetic surgery procedure called Botox? Well, what they do here, well, first of all, let me describe when botulism is bad. So botulism is bad if you get a large dose of it into your blood. Then it can circulate systemically throughout your body and affect all your muscles, and it can potentially kill you. What it would do is it would asphyxiate you. So it'd stop your diaphragm. You wouldn't be able to breathe. Now, the way that people used to die of botulism poisoning, so go ahead and write this down as well, is that back in the early days of canning foods, they would sometimes can foods and they weren't very good at pasteurizing them. So you've heard of the pasteurization process probably. And what that is that's the heating of food before it gets canned so that you kill all the bacteria in it. Well, back in the early days of, when people, used to jar their own foods at home and things like that, sometimes this Clostridium botulinum would get into the food, and then if you let the food sit in its container for months before you consume it, the Clostridium botulinum can actually grow within that environment. So Clostridium botulinum is anaerobic. It does not need oxygen. So in a jar or in a can that Clostridium is in with food, that's a perfect environment for it. The Clostridium would grow, it would release a bunch of botulinum. People would eat that food, that canned food, and then either get very sick or die from it. Now, Botox is different in how it's used. I mean, it's the same substance, but it's different. So in cosmetic surgery, what they do is they take this botulinum poison and they basically just take a little bit of it, and let's use the typical way it's used, and they'll inject it into the muscles around the eyes. And what that does, as you can see here, is it basically causes those eye muscles around the sides of the eyes to not contract anymore. And if they don't contract, usually that procedure will last for, maybe 8 weeks or something like that. If they don't contract during that time, then that actually decreases the wrinkling in the skin around the eyes. And so this is how people prevent wrinkling. And you've heard of, crow's feet or chicken's feet around the eyes when people smile. And so they can use Botox to actually reduce that. And so let me say this, by injecting it into those muscles around the eyes, you're not putting a large amount of it into the bloodstream, it's mostly going into the muscles. And so because of that, it's not going to affect the rest of your body. So Botox would only be bad if you took it and directly injected it into your bloodstream, like in your arm or something like that, which nobody's going to do. And when I say into your arm, I mean into the vessels in your arm. Now, you can also use Botox. There's some other creative ways they've used Botox. So for instance, Botox will also freeze up the glandular cells in your skin so that they don't produce sweat. So for instance, some famous people, before they're going to go on the red carpet, they'll go and get a Botox injection in their armpits so that they don't sweat while they're out in front of the cameras on the red carpet. So there's a number of ways that you can actually use this, and it's perfectly safe to use in that way. You only get really sick if you ingest a large amount of botulinum poison. All right, the other type of toxin is tetanus. This comes from another bacterium called Clostridium tetani, which you can see down here. This is a typical bacteria that's found in soil, and the toxin that this produces is called tetanospasmin, and this causes severe muscle contractions, as you can see here. And so this is a drawing from, I think back in the 17 or 1800s or something like that, of someone who has contracted or gotten Clostridium tetani and that into their blood, and it's creating tetanospasmin, so tetanus, in their blood. And what this does is it circulates around and it causes their muscle cells to contract. Now, this works in a slightly different way. It actually blocks the inhibitory impulses from glycine and GABA. So if you remember, those were inhibitory types of neurotransmitters in the brain. And those two neurotransmitters, in fact, inhibit alpha motor neurons. And so by inhibiting the inhibitor, they actually cause further contraction of muscles. So this causes basically unopposed muscle contractions and rigidity, as you can see in this picture here. This is basically someone who is in full tetanus, and this is oftentimes the way that they would look. All their muscles contract, and because your back muscles are so strong, they actually wind up in this arched position. But you can see how their leg muscles are fully contracted, their arms are fully contracted here as well. I mean, it would just be horrible to have this. And of course, if any of you know about this, of course, we have shots to be able to offset this toxin. But if you were to, for instance, people will get this from stepping on a rusty nail or something like that, then you have to go get the tetanus shots. And in about 10% of cases, in the past at least, getting tetanus can lead to death. So if it's not treated, having this happen to you will eventually kill you. So it can be quite toxic. All right, so with that, that's everything I have today. Thank you very much.